In this primary article, researchers aim to confirm whether the crude venom from the Pelagia noctiluca jellyfish evokes inflammation and oxidative stress processes in rats. Pelagia noctiluca, also known as the purple striped jellyfish, is one jellyfish in particular that scares tourists away from beaches, especially in the Mediterranean. Pelagia noctiluca is one of the most dangerous jellyfish in the Mediterranean Sea, where its blooming has been very abundant for many years. The sting of this jellyfish is venomous to humans, and in cases of allergic reactions, life-threatening conditions can occur. As a general feature, Sandarian toxins may cause damage through two mechanisms of action. They may create pores into the target membrane or induce oxidative stress events. Reactive oxygen species, or ROS, play an important role in inflammation and shock. Temple, a member of the nitroxide compounds family, has been extensively studied in animal models of increased ROS levels and in hypertension. These studies allow Temple to be known as a treatment of inflammation and reduces ROS. Specifically in this experiment, they investigated the effect of Temple on mortality, lung and intestine injury, formation of nitrotyrosine, intercellular adhesion molecule, inducible nitric oxide synthase, and polysynthetase activity. The research questions are as follows. Does crude venom extracted from the jellyfish Pelagia noctiluca elicit inflammation and oxidative stress processes known to be mediated by ROS production in rats? And will a powerful antioxidant such as Temple reduce crude venom-induced inflammatory response? It is hypothesized that, since it is proven that humans with this jellyfish venom injection causes local pain, burning, swelling, hyperpigmentation, and other local symptoms, it can be inferred that inflammation and oxidative stress will occur in the ejected rats. Lastly, it is predicted that crude venom injection will evoke inflammation and oxidative stress processes, and administration of Temple after crude venom challenge will result in a significant reduction of each parameter related to inflammation. The experiment was divided into two steps. In the first one, they initially evaluated the toxic effect of crude venom on blood pressure and mortality. For this purpose, rats were randomly allocated into the following groups listed on the screen. Be sure to note that each group contains 25 rats. As a second step, to confirm the inflammatory effects caused by crude venom injection, they planned to treat injected rats with an antioxidant such as Temple. With this aim, the dose of 30 micrograms per kilograms of venom was used. Rats were randomly allocated into the following groups listed on the screen, also containing 25 rats per group. For blood pressure measurement, the mean arterial blood pressure was continuously recorded and a rectal thermometer was inserted, while rats were kept at a body temperature of 38 degrees Celsius. 6 hours after injection, the animals were euthanized and the organs were removed with a scalpel for observation, while nitrate production was measured in plasma. For histopathological examination, the tissues were also removed with a scalpel after euthanizing. Sections of each type of tissue were evaluated using a scoring system. Molecular and systematic parts of the rat anatomy were observed at the different doses such as blood pressure, pancreatic injury, hepatocellular injury, renal dysfunction, lungs, intestine, and so on. Figure number 1 in the top left corner is the effect of crude venom on blood pressure and mortality. It's graphed with minutes in the x-axis and blood pressure as well as mortality in the two y-axes. All 4 experimental groups are plotted in the two line graphs. Figure number 2 in the bottom right corner is the effect of crude venom on hepatocellular, pancreatic, and renal dysfunction. The second experimental groups are plotted in the bar graphs against the following enzymes in the y-axis: ALT, AST, bilirubin, alkaline phosphate, lipase, amylase, and creatinine. These enzymes were measured in plasma as indicators of the multiple organ injury and dysfunction. Figure number 3 in the bottom left corner is the effect of crude venom on histological evaluation of the lung and intestine. Representative lung and intestine sections from the sham rats demonstrate the normal architecture of these organs. The hypothesis that Pelagia noctiluca venom acts via oxidative stress at the cellular tissue and systematic levels is reinforced. It is concluded that Temple may have a use for treating inflammation conditions as it helped relieve the effects of the venom, which supports the predictions made. It's important to note that administration of Temple prevented all metabolic modifications. In Figure 1, there's a large reduction of blood pressure and an increase of mortality observed at the 30 and 60 micrograms per kilograms doses compared to the 6-unit dose within 1 hour. In Figure 2, injected rats exhibited higher enzyme plasma levels compared to the control group. Findings are consistent with the development of hepatocellular injury. Rats also exhibited higher lipase and amylase levels in the control group, as well as an increase in plasmatic creatinine concentration, which is consistent with the development of pancreatic injury and renal dysfunction. And in Figure 3, lung and intestine biopsies showed marked inflammatory changes with a pronounced cellular infiltration after venom injection. Treatment reduced the pathological changes in the tissues. This study was very intriguing for me, but I'd like to see these research implications further employed to determine how long a subject can wait to receive treatment and still survive after being stung by the Pelagia noctiluca jellyfish.